in previous laboratories, you've gotten experience running Unix commands with bash headers within Google Colaboratory. And you've also used Python within Google Colaboratory as well during the pandas and seaborn units. This time we'll be introducing another programming language, R, and that will also be used in Google Colab. And the reason it's used is because there's a lot of very useful packages out there for bioinformatics that are written in R. So yeah, we'll be using this %sign R header, though before you can use it, you do have to run this little bit of code. It's just a short incantation to essentially enable R within the notebook. As the name would suggest, the command load extension literally loads an extension called rpy2. And what rpy2 does is enable usage of R with the R header. And then you can test that this worked with the version command right here. It's a function in R that prints out information about whatever version of R you're using. Sometimes it can be useful when, let's say, you want to run a certain bioinformatics tool, but it requires certain versions of R, and then you can double check whether or not you have the correct version by looking at the output of this version function. So here we have the major version 4 and minor version 1.2 of R. Okay, so this lab's a bit different. As you notice in the lecture, there wasn't any code, but that doesn't mean we can't still do bioinformatics related things today. Specifically, when you're looking at lots of R packages online, oftentimes there's quite a learning curve to trying out different tools. Thankfully, there's lots of documentation for the R tools out there, and we can access them. One way is through a host of R packages called Bioconductor. So, for example, if we follow this link and go to the Bioconductor page for DESeq2, we can see that here's the name of the package, here's what it does, and as well as some more descriptions and a paper citation. And then a little information about how to install DESeq2, and as well as a bunch of other details. So in order to, oh wait, actually, so since Sergey didn't introduce DESeq2 yet in the lecture, as a short way of describing it, so within the lecture, he introduced using like FASTQ files, and either through alignment to some reference or through quasi-mapping, that you're able to get count data. And by itself, there's some simple arithmetic you can do to that count data in order to compare samples within the same groups or replicates of the same sample. However, these simple arithmetic transformations aren't appropriate when you want to compare gene expression between samples. So in order to do that, you'll want to do something called differential expression analysis. And that involves more complex statistics and essentially through that method, you can now compare different samples. But yeah. anyways, so today we'll be using this DESeq2 tool, and but let's say we're just finding out about this package and just found this web page for the first time. Then in order to find some information about it, we can scroll down to where it says PDF. And if we click on the PDF link, it will take us to a manual. Every R package has this because when people make the R packages, the code that they write is somewhat self-documenting. So they can automatically generate reports such as these. And what's contained in these reports are information about all the functions contained within the package. So if you recall the general idea of functions from the Python module, they're collections of commands that when you call the name of the function, you can run all of those commands at once. For example, the main function in this package is called DESeq. And you can read about it here, about the steps it uses, and then the other functions that this function will call. So yeah, you can have nested functions. But the point is, pretty much anything you need to know about how to use these functions are written here. And yeah, it's very helpful. And whenever you use R packages, you'll probably find yourself using these manuals a lot. So in addition to the manuals, the R community has also produced things called vignettes. And for vignettes, you can click this HTML link right here. And while not all R packages have this, those that do provide kind of a tutorial with lots of code in a kind of notebook fashion even, not completely dissimilar from the notebooks we've been using so far. So then you see here's a table of contents, and there's all these sections we can learn about. And in each section, there's lots of code. And then after some code blocks, I will show the output. So just like Google Colab. In addition, as we're scrolling down in the DESeq2 page, there's also the section for describing the commands you can use to download DESeq2. However, these commands by itself do require some tweaking in order to work in Google Colab. Otherwise, it won't behave particularly well. And to do that, well, first you need the R header. And additionally, you want to add the commands quiet equals true and quiet equals true here, and then ask equals false for the second line. So in the first line, just quiet equals true, and then second line, both of these arguments. Also notice that unlike Python, in R, these true and false type data are written in all capital letters. So if you try to write it with lowercase letters, then you will encounter an error. So that said, while Bioconductor does have lots of great packages on its website, it doesn't have all the R packages out there. And for that, you can download packages directly from CRAN, or the Comprehensive Archive Network, which pretty much has all packages. Though sometimes you'll also find packages that you can get from GitHub. And the way you do that is using this install GitHub command that's part of the the dev tools package. So that's what this double colon syntax means. It means that this string to the right of it is the name of a function from the package that has whose name is represented by the string on the left. In any case, the package that this particular line of code installs comes from a repository called Hadley, and the package itself is called dplyr. Again, you'll want to add the quiet equals true for compatibility with Google Colab. And when you run this, sometimes you might get a prompt. So let me just know.
Excuse me, but DevTools is used to install packages from GitHub.
Yes? In this case. In
this case.
Yes.
Hadley, what does it mean, Hadley? This is the name of a package.
Hadley is the name of the repository. So
dplyr, this is a package.
Yes.
And quiet true to have to be comparable with Google Colab.
Why adding
quiet true. It's quite
okay.
True. It's for what?
So let me show you.
Not show the logs.
So if you do try to run something without quiet equals true, then it'll mess up your notebook and by outputting a whole bunch of text. So okay, that one's fine, I guess, but if I scroll down and move this, for example, then you'll see it just continues to output lots and lots of text. And this will continue to grow while it downloads what it needs to download. And it makes it very hard to scroll through your notebook when you have a lot of packages that you're downloading, or even just a few. So yeah, does that answer your question?
What to do? I run this DevTools install GitHub to upload a dplyr package, but it's running and running.
Okay. You should have a prompt that'll appear after a while, and the prompt will ask you to input a number. I'll show you how to fill that in once my notebook is reset, which I think it has. Okay, so in this case, you'll just type in 1 because it's asking what kind of packages you want to update within this R environment. And you select 1 and then press enter, and then it'll accept that and then continue to download packages.
Yes, it continues to download. I entered 1.
Yes, it'll take a while.
Okay.
Sometimes it can take quite a while. There's a package that will download later. Specifically, DESeq2 takes around 10 to 15 minutes. It's just a really big package. In any case, though, sometimes you'll get this prompt when you try to download from GitHub. Sometimes you won't. And that's okay. Anywho, moving on. So continuing when you're looking for packages or trying to learn more about packages that seem promising for your research, there are these links that you can check out. For example, there's this link in which you can put in the names of different R functions, and it will tell you on the side what packages they're part of. So this install packages function is part of the utils package. And then when you go here, it'll show the same thing you'd see in the manual. However, you don't have to go look up the entire manual for the utils package. You can just search the name of the command, and that's quite handy. At the bottom, if you scroll down far enough, there's examples of how to run the command as well. So we can also look at like, what's this? You can just pick one of these functions in the DESeq2 package. And likewise, it'll provide information about that specific function. Count is probably a common function too. So if you put it in here, you can see that there's actually multiple packages that have the counts function. So this one called like riblom, and then fish methods, and then phyloregion, so on and so on. So yeah, this is a good place to look up functions. Like for example, if you're looking for some code to achieve a certain outcome, then maybe someone posts the code online, but it uses a function that you don't know what it does. Then in that case, you could copy that function, and if it's an R function, paste it here, and you should see some kind of documentation on it. If you don't, then sometimes that happens. And even if the R package exists, it's just because this database isn't completely comprehensive, like the CRAN hosting website is. In addition, there's also this website, which is quite similar to the last website, but it also provides different kinds of information. For example, the search here is different. Instead of showing packages on one side and functions on the other side, it shows you just whatever matches the closest. In this case, there's even 3 functions that are all the same function because they're from the gene filtering package, and this is the gene filtering function. Just because it's posted 3 times on the website. Sometimes you'll also find links to papers here. Or just even useful things like this, like, okay, we know that this repository called Scenic was made by these people. And you wouldn't get that information from this this other database, which is better for looking up functions specifically. But if you want general information about any R packages and various metadata, then this is a good website for that. Anywho, so moving on. So for lab assignment 8, since our programming in R, like we haven't asked anyone to program anything super complicated yet, and we're just introducing R today. So obviously we don't expect you to write your own R code. So instead, what we'll do is, for this assignment, is test comprehension of being able to read these commands and understanding what they're doing. So to start, you'll want to use these resources mentioned earlier in the notebook here to answer the questions that you'll see below, and you'll be assessed on how many of the questions you get right. In addition, keep a lookout for similarities between the R code and Python code you've used already, because sometimes the symbols are the same, and there's also the general ideas that are the same, such as variables. And in that way, you'll be able to read R a lot better, not that if you use your Python knowledge that you know. Anyways, so starting off with running DESeq2, okay, factory resets. I'm going to reload the R extension, and as well as download the package manager for Bioconductor. Because as we saw before, DESeq2 is a package from Bioconductor. And the question here, which I won't answer now, because it's the part of the assignment to answer it, is that what it's asking, what will happen if the above code block is run multiple times, and based on its behavior, how many copies of this code should you have in a Colab notebook? But yeah, you'll answer that in this section that's highlighted in red. It's just a regular text box. You can remove what's there and type your answer. Or I guess if you've fancy red text, you can put it in here and it'll be red. Anyways, moving onwards. Actually, wait, this is, now that we've actually reached the code part, okay. So in a blank notebook, if you'd be running the DESeq2, then you'd start by running this code and then this code, and it'll just chug along. It takes time. This step in particular, as I said, or alluded to earlier, it takes quite a while, and specifically almost 15 minutes to complete. So I'll probably be done explaining the notebook by the time it's finished, but anyways. So then this question 2 is asking what this library function does, and it'll be up to you to look up what that does. So once DESeq2 is downloaded, there is some matrix data that we can put into it. So you can think of this as in the lecture, after either aligning or mapping or using the quasi mapping, you can get count data from that. And this is essentially that count data stored in a file called genematrix.csv or underscore. And so you'll download it from this link. And in general, this is how you download files into R. And then, yeah, so what this code does specifically is that here you have the link to the file, and you'd save it in the variable. Here, this variable has a name count data link. And then using this download.file function, you'd give it the link and you'd give it the name you want the file to be. And then you'd also add quiet, otherwise it'll make a mess. But yeah, there's a couple questions about that, about where the file will get downloaded to, and some questions about what quiet means, which I've already answered. So then after this, we can read in the file into R. So right now with this, it's just this command over here, it's just downloaded into the notebook. But here, we now import it into R. And again, saving it in a variable. This one we call count data. And it's kind of like a data frame in pandas, except data frames are much different in R. In fact, if you try to ask R what data type this is, it'll say it's a list, which it's because lists are also different from Python. But the point is here is that you store it in some kind of variable. And then just like we can access data frames in pandas with the head commands, there's a head function in R as well that you can use to look at the beginning of a file. And here we can see the names of a couple of genes, or rather their accession numbers. And then for different samples, we can get counts of genes. And so then this question here is asking, if instead of a CSV file, what if you wanted to import a TSV file? So one that is tab delimited instead of comma delimited. That might be something you want to do. So helps to know. So in this next part, we're downloading another file, same as we did before, except this file provides metadata. So essentially what it says here is that it tells us the name of the sample and whether that sample was part of the control or the treatment of the hypothetical study that this data is from. In addition, you can access columns in these data structures, similar to how you access columns in Python data frames, except it uses a different syntax. And you can practice it here, where here we're accessing the first column that's called ID. And then here's the data that's in that column. Okay, so moving on. So before we can process things within DESeq2, which is specifically run those advanced statistical normalization methods, we want to first create a special data structure that's unique to DESeq2 called a DESeq dataset. And the way we do that is with this function that's called DESeq dataset from matrix. And as the name suggests, we take in matrix data. So our metadata plus this tabular data we have here, and we combine it into a DESeq dataset, or DDS for short, similar to how in Python we abbreviated data frame as DF. In addition, if you call the name of this special data structure, it won't show a physical representation of what it is, but it will give you some statistics. And that can tell you a bit of its content. Anyways, moving on. You can then use the core function of DESeq2 called DESeq and give it this DESeq dataset data structure. And then what it'll output is still the same data structure. However, it'll have modified its in order to normalize for, as was shown in the lecture, both library size as well as RNA composition. So yeah, if you can go check the slides to review what that is, if you'd like. But anyways, successfully running this will output this output. And then once it stops outputting things, then you know it's done. In addition, do note here that because we use the same variable name on both sides of this expression, we will overwrite the old DESeq dataset data structure. And that's for the most part okay, but in some situations, you may also want to preserve the unprocessed DESeq data structure. In addition, as you may have noticed now, all these arrows here function pretty much the same as Python's equal signs, and that you instead in R see equal signs only for arguments, function arguments. Again, just part of the R syntax, and the more you see of it, the more you'll get used to it. Anyways, so then we can now use another function called results to extract what's called a results table from the DESeq data set. And what the results table contains, as we can check with head, is different statistics for each gene. Because remember, in this case, we ran DESeq2 to compare differential expression between our control and our treatment. So yeah, each of these also means different things and their abbreviations. You can look that up in the documentation. It's good practice. But yeah, continuing on, you can also get summary statistics for each of these columns, similar to the describe function in Python, where it would give you this kind of information for a data frame. So that this results table pretty much summarizes the results of the differential expression analysis. However, the numbers are pretty hard to look at. So there's ways we can visualize the DESeq2 data. One way is with what's called a volcano plot. And as you'll see later on in the lecture tomorrow, as Sergey continues that, you'll see that does it show? Nope. Okay. This. Okay. This code on the right is still buffering, so I'll just show you what a volcano plot looks like. So it comes in the shape, and on one axis, you'll have the log fold change, which on to the right, typically you'll have the genes that are like highly expressed relative to the control. And then on the left side, you'll have genes that are underexpressed relative to the control. And the y-axis is the p-value. So remember that there's lots of statistics that go on in DESeq2, and those statistics will output a p-value. So yeah, typically it's also common to see parts of the volcano plot grayed out because those points are not very important, because we define a certain threshold and we are only interested in the points that are beyond that threshold. Sometimes multiple thresholds can be defined too, where in this case, for example, oh God, that's blurry. Like here, there's both a y-axis threshold and two y-axis thresholds and one x-axis threshold. Okay, cool. The DESeq2 finished installing, so I can actually show you the volcano plot that we can make from our results table. Count data. Okay. So again, importing the metadata that essentially labels which samples are which, whether they're control or treatment. Then we convert this matrix data into the special DESeq2 data structure. And then we run the statistics on this data structure. Then we extract a results table from this data structure. And as shown before, you can get summary statistics of the results table. Next, we want to install this package called Enhanced Volcano from GitHub. As you'll see, this is an example of a GitHub package that actually doesn't ask for a prompt. So you don't have to worry about running it. Anyways, so then once that finishes, which it just did, we can use this package, specifically this enhanced volcano function within the package, to plot a volcano plot. So once you get the check mark, that means it's plotted. And you can check in the left-hand screen, and then you can download the graph. So here's what it looks like, where we're only really interested in the red points. The blue points and the green points and the gray points don't meet our thresholds that we've defined.
Are you showing something? Because we only see the notebook. That's the question.
Okay, I see. Sorry about that. Okay. I see. That wasn't good because I was running code on the right.
Well, now we see.
Yeah. Whoops. Yeah. But anyways, here are these red points, and this is what we consider to be both statistically significant and significant in terms of fold change. That is, there's enough change in expression that we care about it. Like these blue points, for example, They are statistically significant, but it's such a small change that we don't care about it. Anyways, moving on, there's some questions about this that you can answer, both by looking at the graph itself and looking up information about the enhanced Volcano package, I think.
Cynthia, may I have a question? Do you expect us to give an additional code to find to answer this question, or we have to analyze the picture only? The plot.
I think you don't have to provide any code here. You can just write out your answer with text. And the only question that takes code is this one right here, where you can practice selecting the other column, the status column from this data structure.
Okay, thank you.
Yeah, but yeah. Moving on. There's other ways we can visualize our data too. One of them is a principal component analysis. And to start, we'll want to perform some normalization on our data just specifically for Pca. And then we can use Pca to look at our data set, in which we can see that the controls are very different from the treatments, and that there's like this cluster, or rather, there's 3 points that cluster here, and then 3 points that cluster here, and they're far away from their counterparts within the same sample. In addition, you can notice that this is commented out right here, and you can run this extra line of code, and instead of outputting it to the notebook itself, like with the previous example, it'll provide a version that you can download as a PDF. And the reason you do this is because it allows you to change the width and height of your plots. Pretty much, well, at least all the plots in this lab, but it should be generally applicable to most R plots too, unless the plots are made a very specific way. In any case though, moving on, there's more data visualization we can do. It'll take more R packages. So that's going to download there. And then here, what we're doing here is we're looking at this data structure again, abbreviated as DST. And we're selecting a specific number of samples from there. Specifically, we want the samples that have the highest variance because it makes it easier to visualize when you can have many samples and genes. Anyways, if you run that code and then you install some more packages, once that's done, you can then use this function, heatmap.to, and with this, you can produce a heat map, in which the samples are hierarchically clustered so that based on the expression, they're clustered together based on similarity. And here we can see that all the treatment shown in green clustered together, and then all the control was shown in gray right here, all clustered together. And additionally, we can see that there's six samples, 6 genes right here that they're very, very, very highly expressed in the sample, but not at all in the control. Sorry, not I meant treatment, but the highly expressed in the treatment, not in the control. And that corresponds to these dots right here. And so that's another way of representing this data that you can see as a kind of rectangular pattern here, what is represented as dots in the volcano plot. And then, so yeah, after this, there'll be a couple more questions. And with that, and once you've answered all the questions, you'll be done with the assignment.
I'm sorry, but can we check what packages are already uploaded? So I understand that we need to upload just once any package, and but is there a possibility to check what packages are uploaded at this moment?
I know that there's a pretty easy way to do it within our downloadable environment called RStudio. As for Google Colab, I don't know off the top of my head, but I can see if I can look something up real quick.
Session info. If we use in session info, it also works in Google Colab, and it we can see all the packages.
Yes. I just found a little bit of code that you can copy paste. I'll screen share again real quick. Yeah, if you copy this code, then it'll list out all the packages. You might notice that you didn't download all of these individually, and that's because when you download certain packages, often those packages will rely on other packages, and those are known as dependencies, and they'll get downloaded alongside whatever packages you're interested in.
Sorry. So when we want to download, yes, to install a package from Bioconductor, Bioc Manager, this is a function, or this is the name of, what is this Bioc Manager? This is the, yes, I just ran the code, but I know what does it mean, Bioc Manager. This is the name of the package.
Yes, it is the name of the package, the Bioc Manager package, specific to Bioconductor. So Bioc is short for Bioconductor, and they have their own package manager because they prefer doing things their own way, as opposed to the way that you download packages from CRAN. Supposedly it's better.
Can I use here just as in R install packages, install.packages, and then the name of the package?
You can. That'll install it from CRAN instead. If you do that, do also add the quiet equals true. Otherwise, they'll make your console messy. But yeah, you can do that.
It's possible also to do.
Yes.
Okay. Yeah, thank you.
Yeah.
When you ask in the question 2 for the documentation for library function, you mean any documentation from any website, like the one you showed before, or documentation.org?
Yes.
Mhm.
Just anything that describes what the function does.
Okay.
And like, basically the idea is as long as you can understand what this function does from that source, then it's a good source.
Mhm.
Because
Okay.
this is like supposed to simulate like you found a package for your research and then it has some function and you're trying to figure out what the function does.
Got it. Thanks.
Yep.
Can I ask a question?
Sure thing.
I can't find any information about ask as a parameter. I see there is a function ask. It's like a prompt, but not as a parameter. So just say that it will not change nothing. Will change nothing.
What as a parameter? Repeat.
Ask.
Ask.
Ask anything.
I'm not quite sure what command you're referring to at the moment. Would you mind screen sharing? And then
Download file. It's function download file.
download file. Okay. Let's see.
Question 4.
Thank you. So the ask parameter only appears in the when you install the bioconductor manager. All the other commands just use quiet equals true.
Yeah, but I don't understand what because download file doesn't have such argument, no?
Yes.
So ask is for asking to install or not. It's like interactive form of installing, no? Okay, so I got it. Thank you.
Yeah. But yeah, it'll end up just making your notebook messy since Google Colab is not meant to be interactive like that as much as a more traditional terminal is, or even the like RStudio environment. That said, it does have its advantages in being able to integrate all these different languages. So, but yeah, I hope that answers your question.
Thank you.
Can I ask about count data? Yes, gene metrics.
Sure.
So we have here the first column. This is the name of the gene, I understand.
Yes, the accession.
Yes, this is the individual ID of the gene, yes. And then we have sample 1, sample 2. And the counts, what does it mean? The numbers for each gene by samples.
Yes. So that would be the sequencing depth of each gene. So
what does it mean? I'm sorry, because I'm I'm a bit far from biology. What does it mean, sequencing depth?
no worries.
The number of reads, what?
Let me see if I can bring lecture slides up.
To look at the lecture?
Yes.
Okay, I will have a look at it.
No. I'll bring it. I'll bring it right now so I can explain. So, okay. There's a lot going on this screen, but to simplify it, just looking at this part right here. Shape format, just this part right here. So you can, looking at this sample, you can see that this gene, this green gene here at the bottom, was expressed a lot more than this red gene right here. And so if you think about a complete row of reads that connect together in a single line, that would be one fold coverage. And so here, maybe at the top for the red gene, maybe you have like 1, 2, 3, 4, 5 fold coverage. And then here you'd have like many times that, maybe 20, 30, 40, 50 fold coverage. And so this is what sequencing depth refers to, and what the numbers in the count data refer to, as in like you have at least that many copies of that gene that were expressed as mRNA.
The degree of expression, we say, yes, of a
Yes.
gene. Yes, the degree of expression. So gene DE has a higher sequencing depth, you say, yes?
Yeah.
Than GZ.
Had more coverage.
Okay, I see. So this count means the sequencing depth for each gene in different samples.
Repeat that.
So these numbers, yes, I come back to this data frame. Yes.
Yeah.
Count so gene matrix. Yes, for each gene, for example, in sample 1, for the first gene, yes, we have 723. So this is the sequencing depth of this gene.
Yeah. Probably in that case, the better term for it would be coverage in this case, because depth can also refer to depth of sequencing in libraries. So but that's a different thing. But the point is coverage is probably the best term there. Yes.
Okay. Okay, I understand. Yes, more or less. Thank you.
Yeah. Okay. Anything else?
No, meanwhile, this one. Just to understand what is about the data.
Yeah.
Thank you.
Happy to help.
I'm sorry. And the number of genes in a gene matrix data, yes, I can see from the output of DESeq data set from matrix, yes, function. And I understand it's also if I count the unique values in the first column, because the first column, yes, this is the ID of a gene.
Yes.
Yes, it would be the same, yes? The unique values in the first column.
I believe so.
Yes, the first, yes, I looked, it's the same. I just wanted to be assured that the first column, this is the name of a gene. Yes, and yes. The question, what is the number of genes? We need to know, so the number of unique columns.
Yes.
Yes, and or from this, this function, DESeq data set from matrix, gives you some outputs, yes, that provide us with this information, right?
Right.
I understand correct. Okay, thank you. Sorry, when the question 8, what does it mean? The unprocessed DESeq data set data structure. It means we need to add an argument to DESeq function to prevent this long output.
Not quite. In this situation, recall that I said that the DESeq function will process the previously made DESeq dataset object.
Yes. Yes.
And and by the end of it, it will have a normalized version of it that's processed. But if you want to preserve the unprocessed version, that means you have to save a copy of the original version somehow. I guess
we don't need to overwrite it.
No.
Yeah, that is why we need to make another object and okay.
Yeah, you can use variables for that.
Yes, another variable, and then to give a function to another variable.
Yeah. Yeah, and if you do that before you run DESeq, then you'll have a version of the original and then a version of after DESeq.
Okay, that's right.
Yep.
Thank you.
Yeah. So for today's lab, it's a bit of an extension of the previous lab. So in the previous lab, we showed some code for running the DESeq2 tool. And to help make that easier, we provided some matrix data at the beginning. That the matrix format is one of the formats that's DESeq2 will readily accept. And let me just make sure the link is right. It is one of the easy formats that it'll accept. And however, sometimes it can be hard to get actual data from a pipeline into the matrix format. Other times, you may not want to use the matrix format itself because there are some studies which suggest that instead of using direct counts that are contained in the matrix format, it's better to use other formats. One of those formats is Quant Sf File that's output by software called Salmon, which was mentioned briefly by Sergey before and is also in the lecture slides. What Salmon is it's a quasi-mapper. So unlike aligning tools, which seek to count genes and transcripts by aligning reads to those genes and transcripts, a quasi-mapper will instead develop a profile based on statistical characteristics of the reads that you have. And it will compare that profile to the profiles, the statistical profiles of the genes themselves, and there, in that sense, get counts for those transcripts / genes. And so what the quant.sf file looks like is this format. So on the left hand column, we have an ensemble ID for transcripts. And in the next column, we have the length of the transcript, and then effective length, which is similar to that. We have transcripts per million. That's a measure of a normalized measure of the relative abundance of transcripts with respect to one another, and within samples, not across samples. And then also a number of reads here. One thing you'll notice is that these are actually fractions, and part of the reason for that is because if you recall, the Salmon doesn't align reads specifically to a genome or a transcriptome, so it doesn't have exact number of reads. Instead, it uses statistics to compare profiles. And based on those statistics, it generates a sort of theoretical number of reads, which itself isn't a whole number. In any case, in order to import this kind of data from salmon into DESeq2, as we've seen before, we will use either one of two packages, tx import or txi meta. We'll use txi meta here because it's a bit easier than tx import. The reason for that is because tx import also requires an additional file that you'd have to generate yourself, and that requires writing a fair amount of code. And we aren't, as a class as a whole, we aren't quite at the point of writing our own code for this stuff yet. But that is an eventual goal, because in order to use these tools, it helps a lot when you know how to manage errors by writing your own either Python, R, or Unix scripts to deal with those errors. Anyways, go on.
Is it possible some questions about the structure of the data? So these lengths and effective lengths. What does it mean? Yes, this is a length of, I understand, of a gene and how it is expressed. Yes, and transcript per million of what? Yes, I'm sorry, I'm not have a very solid background. That is why I just simple. Yes, I'm sorry.
So length itself refers to the length of the transcript. Effective length, I'd have to double check, but if you notice, all the effective length numbers are smaller than the actual length of the transcript. And that probably refers to how many nucleotides were covered by the reads. Because as you imagine, when you're sequencing, sometimes you won't get all of the nucleotides within a known transcript. Does that make sense?
Yeah, so this is a number of nucleotides in a gene, yes, and
I believe
effective,
so.
Yes, effective lengths, I don't know, maybe after exclusion of introns, something like that. So another, which is in fact close to the length, to the second column.
Yes.
Okay, yeah, thank you. And this per million of,
so
per million of what?
So the
what
per
is
million, okay, so transcripts per million is a method of normalizing your count data within samples. So let's say you have different runs of a sample on the sequencing machine. Sometimes for technical reasons, one of the runs gets lots and lots and lots of reads. And then one of the other runs doesn't get that many reads. They're still from the same sample. So relative amounts of gene expression between those samples should be the same. However, the absolute number of reads would be different simply because more sequencing was done on one of the runs. Let me see if I can bring up a picture of that. This one.
This is what we said yesterday about sequencing depth. So it's called something like that.
Yes.
Yeah, sequencing depth. And this TPM is just a relative measure. Yes, it can be used in fact for comparison. Right?
Yes.
Between the genes.
Yes, and the million itself, the million does not have a unit. It's simply just the standard million number that you just divide by in order to normalize for sequencing depth.
It's like a normalized measure, like standardized measure.
Correct.
Okay. Okay.
So like in this slide, if my computer will stop lagging. So see both of these, just pretend they're from the same sample but different runs. you'd expect the same amount of relative expression, which you do see. All of these genes shown in blue are expressed about the same amount on both on the left side and the right side. However, just because the left side was sequenced more, it has greater sequencing depth and hence more reads associated with it. However, that doesn't mean that these genes were expressed more on the left than on the right. So those first two columns describe the lengths of the transcript, and then the TPM and number of reads describes the expression. Anyways, so as I mentioned before, TX import is used to convert these transcript based abundances into counts that will be used with DESeq2. There's some research supporting that this is the best practice, and you can read about it from this paper, which was made by the same people who developed TXI import or TX import. But yeah, that's additional reading. And anyways, moving on.
But where's the sample? Like we only see one sample, or this is the profile of all the samples?
This here is just one sample. So
Okay,
every Quant Sf file is a single sample.
Oh, okay. So we'd have to import like a bunch of samples for which to be relevant.
Okay. Anything else before we move on to the assignments? Okay. So then moving on. So like I mentioned before, we're not quite ready yet to write our own code from scratch or near scratch. So in this case, what we'll be doing today is using DESeq2 along with TX imports. And instead of answering questions about the code last time, you'll be filling in blanks represented by this black square. And those blanks are important aspects of the code that it's important that you get them correct. Otherwise, the code will not work properly. And being able to fill in those bits of code, if you are able to do it, then that means you know a bit of what's going on. And that's important whenever you're running code, not to just blindly run code that you've seen somewhere else. But anyways, so the goal of this assignment will be to have all the plots that were plotted last time also be plotted this time. However, like I mentioned this time, there's bits of code that you need to fill in. Okay. The data that we'll be using for this experiment itself comes from this study. It's a pretty popular sample data set used for teaching differential expression methods. But essentially, the study is looking at asthma and patients that underwent glucocorticoid therapy. And so the treatment in this case are patients who went through that therapy, and the control are people who didn't. You can read more about this in this link that I posted in chat, which I'll also post in Slack. But yeah. Okay. So the overall theme is the same as before, in that you load in the Pi2 extension in order to enable R within the notebook. Then, because there are some packages that we want to get from Bioconductor, we also run this bit of code. So then the packages we want to install are both the TXI meta package, as well as the DESeq2 package. And here's most of the code to run that. Moving on. So in order to access the data from this study, one of the places you can access it is their GitHub repository. And so we'll use the knowledge that we learned in the GitHub lesson to clone this repository into Google Colab. So essentially make an entire copy of the repository inside our folder here. And you can take a look at this repository and then get an idea of its structure. What's important are the Quant.sf files. So here it says quants, and then here are different SRA run session numbers, and then here's like Quant Sf files. In addition, you can look at those Quant Sf files since they are, as we saw before, in a kind of tabular format. We can use pandas to look at them. And so a bit of a throwback, but using pandas, you can get a quick idea of what the data looks like, as well as some summary statistics. You could go further with this if you so chose. For example, plotting like distributions of lengths or distribution of transcripts per million to get a sense of your data. Any of the data wrangling or data visualization that we've done in the past can be done with this data too, because it is tabular data. So yeah. Next, there's a file called SRA run table.txt that you'll also want to check out. It's in this repository. You can go look for it. And you want to import it with as an R variable. So as was mentioned in the lecture, probably in R, this assignment symbol with the arrow is equivalent to the equal sign in Python. And for that, we can save the output of functions, such as this read.csv function, into a variable. And then whenever we call the name of the variable, we can access the contents of the variable. In this case, the SRA run table.txt file. So moving on. One of the things we need to get is the absolute path of every quant.sf file. So the reason for that is because there are multiple samples within this repository. And if you check the SRA run table.txt file, you can see that different runs will have a different treatment. So the pattern is untreated, and then treated, and then untreated, then treated, then untreated, then treated, and so on. So yeah, because we want to run differential expression analysis on all these files, we need to know their locations, and that's why we need the absolute paths. The reason for that is also because what TXI meta does is it actually uses more than just the quant.s files itself. It also uses these other files, especially the .json files, to help make the import process easier, which is, again, why we're not using TX import. We're using TXI meta. Okay, next, we want to create an R data frame containing these absolute paths, the SRA run IDs, and then treatment status. While it wasn't exactly covered how to make a R data frame compared to like a Python data frame, you don't have to worry about that because the code necessary for that is here. What you need to fill in, though, is the information in order to access the SRA run IDs and the treatment status. So yeah, moving on, we'll then use TXI meta's core function in order to convert the transcript level abundances to count matrices. So again, as an intermediate step between SEMN and DESeq2. This argument here is blocked out. You'll want to look up some information about the TXI meta function in its documentation, and that should help you figure out what to put right here. Then running that, it'll do a whole bunch of downloading because one of the things TXI meta does is also downloads metadata. Again, it makes the whole process easier. So yeah. Additionally, you'll notice that the export of or rather the output of the TXI meta function is saved in this variable named summarized experiment. And the reason why I named it summarized experiment is because it's actually the name of the data type it is too. There's a special data type called summarized experiment that is used a lot within DESeq2. It helps just make things compatible. In fact, in the last lab, we also dealt with summarized experiments at certain points, specifically wherever you saw, what's it called? Wherever you saw the assay function, that's where there's a summarized experiment. But anyways, if you want to learn more about this special data type, you can follow this link and read about its format. Then that might help make some of the other functions clear as to what they do. Anyways, like the DESeq dataset function or DSet dataset object, calling the name of this special data structure will also provide information about it. And this information is important because it'll tell you information about rows and the data in those rows, as well as columns and names of the columns. So yeah, then we'll want to import the summarized experiment into DESeq2 with the DESeq dataset function. This is essentially the equivalent of in the previous lab where we used DESeq data from matrix function, except because, as the name suggests, that function is used for importing matrix data, whereas this function is more of a general function that works with the output of TXI import and TXI meta. Anyways, one of the arguments is this design argument, and this is a pretty important argument because it will vary depending on the data that you use and what things are named previously. So you'll want to look up information in the documentation for this function and see what it says about the design, and then using the information in this part, pick the proper name to put there to get things working. In any case, by importing things into DESeq2, you'll produce the DESeq data set object or data structure, which you've been familiar with before. And previously, we'll process it with the core DESeq2 function, and then we'll extract a results table from that object. And with this results table, we can then plot a Volcano plot. First, for that, we want to import our plotting program called Enhanced Volcano. And then we'll want to plot the Enhanced Volcano, the plot with this function right here. And what that will do is it'll give you a plot that looks like this. You'll actually notice that this plot kind of has the same shape as the same one as in this study. So figure A, it's kind of similar. But yeah, the reason for that is because it's from this same data set mostly. And definitely from the same study. And anyways, so that's for the Volcano plot. And then next for the Pca plot, as before, we'll use this VST function to transform the data and produce yet another special kind of data structure called a DESeq transform. And then using that, we can produce the Pca plot. This also has a special argument called int group, and you'll want to look up the plot Pca documentation for this as well. And like with the design argument right here, this int group argument is also pretty important because it'll determine whether or not you get an error or you get a plot. And potentially, you can make different kinds of plots with this argument, depending on, well, you'll read more about it when you look up the documentation for plot Pca. Finally, there's the heat map. And so here are packages that we want to install, pretty standard. And then we want to select a certain number of genes with the highest variance between samples, because we don't want to look at all every single gene, because there's thousands of them and that'd create a pretty insane heat map. And so we just want the highest expression ones. Anyways, with that, we'll have the heat map here, and this is the code for it. There's this bit here, which is also blocked out. That's something else you need to figure out what to fill in. And yeah, so yeah, that's the assignment for assignment 9. Depending on your prior knowledge with R, this may be a very, very easy assignment, or it can be kind of hard to figure things out as well. So if you find yourself wanting more after this, then there's also an optional challenge where if you know enough R, then you should be able to write your own R code. And in that case, you can check out the Biomart package. And what this package will do is it will look up the ensemble transcript IDs through Biomart. For example, where's the... But okay, yeah, I'll look it up on this website, and for all of the thousands and thousands of transcripts, and it'll then provide the name of the gene that the transcript is associated with. This is something you'd probably want to do if for publishing like these kind of data sets, because ultimately the transcript IDs don't really mean much to anyone. And even if you're just like the exploratory phase, it won't mean much to you either. So you'd rather know what genes these are associated with, because you want to know the expressions of genes, not transcripts usually. And so then you'd convert these IDs with the Biomart package and then remake the Volcano plot with the gene names as labels instead of the transcript IDs.
Excuse me, Cynthia.
Sure thing.
I tried to start this block manager, whereas download and install the 3x meta package, and
Yes.
it shown the parsing error appear here. It's not. Do you see my screen?
Yes.
So here, sorry, here this I tried to install this. So it's 3x meta package. What is happened? It's not.
Okay, so the reason why this code block isn't running is because you need to fill in the parts with the black box, or in this case is the question mark box symbol.
So I should change here some.
Yes, specifically the parts that has the boxes.
This thing
Yes.
I should replace to put here. I understood.
Yes.
So this was the problem, and I think here also.
Yes.
We should replace this. Okay, thank you. I understand.
Yeah. No worries.
I'll try.
Happy to help.
There's an example of how to do just one, but to combine them, we have to do some kind of AND or like pipeline. No pipeline. Like.
Yeah. For that one, you use the paste function.
Yeah.
You could look up a bit of information about paste. It's one of the base R functions. But in general, it's pretty similar to the cat command in Unix, where you can concatenate strings. And so if you look at the arguments to that command, you'll see that and then also look at the output, you'll see that the only thing that's missing is the names of every run. So you'll need to extract from the metadata file the run names, and then the paste function will generate the paths for each of those files.
Okay, I'll try.
Okay, let me know if you have any more questions.
What quantifier did we use for this one? Did we use salmon?
Yes, that's how we generated the Quant.sf files. By we, I mean the people of the study who published this data. I've personally run Salmon myself, and it also produces the same kind of Quant.sf files.
But the type of data, when I indicate salmon, it does not work.
That's odd. It should work because you're on the TXI meta step, right?
Yes.
Yeah. It may be capitalization. How did you write?
No, lowercase.
It may be something upstream then, potentially. Would you like to share your screen?
Yeah, sure. So this
Okay.
is the error I get. Let me just quickly.
Okay. Error entanglement. The files do not exist at the location specified by column data file. So that means there's probably an issue with the path. You'll want to check the stuff that you wrote in the previous code blocks then.
Yeah.
Yeah.
This looked exactly like the assignment result.
Scroll back down to the error then.
Yeah.
Let's see if we can get any other information from here. That seems just to be it.
Maybe I should run.
The error thinks that it's an issue with the file location. Sometimes the errors, it gives programs will give out errors that don't exactly match what the problem is. Oh, scroll up. Scroll. Quantus. Okay. So the reason is that if you look at the extension for or rather the absolute path, and if you look, it says SRS. If you check the files in the left-hand the context window, if you go down the file path, you'll see that the files start with SRR, not SRS. And so it's looking for SRS folders, but they don't exist.
But why?
Because you accessed the wrong column of the metadata file.
Oh, SRR.
Yep.
SRR.
Okay.
Huh.
Yeah.
But there isn't any column with SRR. Oh, there is. Okay, never mind. Okay, thanks.
Yeah. Yeah, see if you can fix that. And if you have any other questions, you can screen share again.
Okay, thanks. Yeah, it worked, but it asked me for a selection if I want to use default or use temp.
Oh yeah, you just have to go through that.
Should I use
selection
default?
doesn't like it'll ask for a yes or a no. You just pick either yes or no. It doesn't really matter. And then after that, it'll ask for selection. So that's like 1, 2, 3, 4, or 5 or whatever. It shows in above a bunch of numbers, like 1, 2, 3, 4, 5 or something. Just pick one of those.
Sorry. And when we run DESeq2 core function on the DDS data, do we need to keep at first DDS data as another variable to prevent overwriting class like we did last time?
You do not need to do that. That's optional. So for that step, you can, in this case, because the name of the variable is already decided, that just means you overwrite it.
Okay.
But yes, like before, if you wanted to save a copy of the DESeq dataset object, then you save it in its own variable, like maybe DESeq2.
Okay, thank you. Thank you.
Yep.
But it's fine to override it, right? For the assignment.
Yeah.
I have a question. When
Sure
we
thing.
create this variable top var genes, yes. So I understand we, as a result, we select the number of rows for those genes where the variety is the highest. The first 25 genes, because
Yes.
the output is so the output, this is so this is the number of the row number. Yes.
Yeah. The genes with the highest variance will be stored within the top var genes variable.
Yes, it means that in this DST data, yes, so the genes with the highest rows will be in a row. So, for example, the first value is 178,460. This is the number of a row in this DST data, right? So this is a row number. And assay function, this function just make DST data looks like a data frame. I understood like that.
Yes. Yeah.
It transforms somehow this, because when I look at DST, it's like a summarizing output. But this assay, when I give this function to DST, I create such like a data frame, something.
Yes. The reason for that is because the DST object is pretty similar to the summarized experiment described earlier. And so let me see if I can get the picture. So you can think of the DST as a kind of summarized experiment object. And so when you use the assay function, what you're looking at is this red part of the summarized experiment that's visualized. So as it's described here, the assay or pink block contains a matrix of counts. And as you look in that in the assignment, it orders those counts based off of expression level, and it picks the 25 highest.
Yes, I have seen the first 25. Yep. The most. So these are the first 25 genes with the highest variability.
Yes.
Yes, with the highest expression. So what would be at the top of a Volcano plot? These red dots at the they will be the highest dots. Or maybe I'm confusing.
They'll be the most widest dots. So the ones with the greatest fold change between the treatment and the control.
Which it will be? See.
It'll look at fold change. Instead of the highest dots, which are the most statistically significant, it'll look at the most variable transcripts. So the transcripts that showed the greatest difference between the control and the treatment.
Yes, so for these genes, how do you call this variable for fold, a double fold, or how it's called? Yes, I think I understood. So with the highest variance,
Yeah,
they will be.
I think they might actually be these dots over here.
Yes, they will be here. Yes, thank you.
Yep.
Yes, it's maybe how to say, it's maybe not so difficult to complete technically this task, but it's much more difficult to understand how the data is processed inside this function, especially when you don't have confident knowledge in biology. But we try. Yeah, thank you.
Yeah, happy to help.
I'm not quite sure that I understand correctly the heat map explanation, how to interpret the heat map, if it's possible.
Okay.
What we produced, yes, because I understood how to did it, but I'm hesitating about its interpretation.
Okay. I'd have to rerun the code to get a larger resolution version, but the version that is in the notebook will have to suffice for now, even though it's a bit on the blurry side. So to explain what's going on, for starters, there's the, oops. At the bottom, oh, I see. Part of it's clipped off. So at the bottom, we have the run names. So you can think of them as the samples, in which the green ones are the shown near the green bar over here. And then over here, those ones are the treatment. And then the gray ones are the control. And so the samples on this bottom part, and then on the vertical axis, it has the names of the 25 most variable transcripts in terms of expression and fold change.
So this on the Volcano plot, so we decided that these are red dots on the extremes of the horizontal axis. So okay, the most with the highest, can I say the genes that have the highest expression, or maybe the highest variability in something, but in something I can't understand.
Yeah. Not necessarily the highest expression, because the ones on the left actually have very low expression, but relative to the control.
To the control.
Yes.
Yeah. So we selected these 25 genes. Yes. And okay. And why some of them are, so we have now different colors, and I understand that these colors, so they in the upper left corner we have this small picture, yes, from the lowest row Z score to the highest, yes, and in the middle. And so I see that so if on the left, yes, the first 3 samples, yes, they have higher row Z scores for the upper genes. I don't know even how to make interpretation. Yes, I see that the first 3 samples, yes, I think they differ somehow from the other samples.
Yes. So looking at, they're clustered based on their overall the overall fold change for all these 25 transcripts. And So the samples that are most similar to each other will cluster together, and those that are different from each other will cluster separately. So for the most part, it looks like the control samples cluster together on this left side, with just one of them clustering with the treatment. And likewise, we can see that the treatment samples cluster together as well. And of course, this could be reordered, and you could also add additional clustering of the samples, sorry, of the transcripts. However, looking at it, just eyeballing it how it is at the moment, you can tell that there's like more red for these top transcripts for the control, and then also here too a bit. But yeah, it doesn't look like there's a super strong signal aside from these top transcripts showing up as red within the control and then blue within the treatment.
But this control, which is between two green, yes. So here's the third sample on the right. Yes, the third sample on the right, because vertically, the situation here is almost the same as for other controls, for the first three on the left. Why this control is here between two greens? Because I would put it together with the other controls, or they are not sorted by, the data are not ordered by samples.
So the reason why it's appearing here is because overall, it's closer in terms of expression to for these top 25 transcripts. It's closer to the treatment samples
to the greens
overall. So even though it's red on some parts, overall it's closer to the green than to the other controls.
And at the top we have this. So these are two clusters. Yes, I understand two clusters, but inside each cluster we have other subclusters.
Yes.
Yes. So for controls we can still detect two subclusters, right? That have some difference between controls. Yes, and for let's say for another cluster which has not controls, but how do you call them, for treated?
Yeah.
For cases and one control. Yes, here inside we have also 4 subclusters. Yep. Like that. So we need to interpret some data. But this histogram, it refers to what?
The histogram in the top left?
Yes. Here we have an histogram, some distribution
of. It's hard to read, but that says count. So that refers to the number of like how to put it, the
number of reads we have.
Yeah.
The number of reads, but they are standardized somehow, I think.
Yeah.
And that score. So if it looks like the normal distribution, maybe with some where we have -1. Okay.
Yeah.
Okay, thank you, Cynthia.
Yeah. In general, there's more packages for plotting expression data than just the 3 that have been introduced so far. So when it comes down to picking them, you'll have to look through the documentation for the specific details of what all the things in the plots mean. And how they generate that, and then you can make an informed decision about whether you want that kind of particular data visualization in your paper.
I see. Yeah, thank you very much.
Happy to help.